Hello. Today we're going to talk about taste, also known as gustation. Each human tongue contains hundreds of small bumps called papillae. The tongue contains 3 different types of papillae that reside in different regions of the tongue as shown. First, circumvallate papillae are located in the back of the tongue, foliate papillae are located on the sides, and fungiform papillae are located on the tip and the middle of the tongue. So each papillae contains small bundles of cells known as taste buds, and these are represented by the yellow dots in the diagram. Now, let's take a closer look at one of these taste buds. To begin, each taste bud is embedded in the epithelium of the tongue, shown on the left and right side. The actual taste bud is comprised of 50 to 100 taste cells, and these taste cells are continuously replaced by basal stem cells located at the base of the taste bud. At the surface of each taste bud, there are small openings called taste pores. These taste pores allow microvilli to make contact with food chemicals that are dissolved in our saliva to reach the sensory taste cells. So the food we eat contains a wide range of chemicals called tastins that bind to different receptors on microvilli. Once the specific tastins bind to certain taste cell receptors, an action potential is created, which then sends signals down the gustatory afferent neurons, eventually leading to the brain. With that being said, each taste cell responds to a specific type of taste. And these types of tastes are sweet, bitter, umami, salty, and sour. So each type of taste has its own unique set of receptors. However, sweet, bitter, and umami all have similar transduction mechanisms that utilize G-protein coupled receptors. Salty tastes are detected by sodium channels, while sour tastes are detected by proton channels. Now, let's take a closer look at how GPCR signaling works to detect a sweet taste. So each transduction pathway has its own unique receptors. Sweet tastes are detected by TAS1R2 and R3 receptor proteins, and when the right tastant ligand binds to these receptors, it triggers the activation of a G protein cascade. Upon activation, GDP gets replaced by GTP. This triggers the separation of the alpha subunit and beta gamma subunits from the receptors. The alpha subunit then activates phospholipase C beta 2 protein, which activates PIP2. PIP2 is then cleaved into DAG and IP3. So IP3 then moves to the calcium ion channels located inside the taste cells, which open ion channels releasing calcium ions into the intracellular space. These calcium ions, along with DAG, bind to and open sodium channels on the cell membrane. An opening of these channels allows sodium to flow into the cell, triggering an action potential. So unlike the GPCR pathways used for sweet, bitter, and umami, salty tastes only use a sodium channel. With salty foods having a high concentration of sodium ions, these ions pass through these channels, triggering an action potential. Sour foods, however, contain a high concentration of protons. These protons pass through proton channels, which change the membrane potential of the cell. This triggers the opening of voltage-gated sodium channels, which allow sodium ions to enter the cell and therefore trigger an action potential. Looking back at the taste bud, each taste cell has one of these specialized mechanisms to detect one of the 5 tastes. So cells that detect the same taste have axons that get bundled together and remain separate from the other taste cell axons, and this is known as the labeled line system. And here we can see that these bundles remain together all the way to the gustatory cortex in the brain. Each axon bundles then synapses to a specific region of the gustatory cortex, which allows for the specific sensation of each type of taste. That's all. Thanks for watching.